Hello and welcome to the ISTH Congress Daily News. My name is Tita Joy Jiasakun. I'm a hematologist at King Chulalongkorn Memorial Hospital in Bangkok, Thailand. Today, I'm very happy to be joined by Dr. Wei Kai Lee from Washington University School of Medicine in St. Louis. Today, he will be presenting to us his abstract entitled Molecular Basis of vitamin K-driven gamma-carboxylation regulating hemostasis at membrane interface. So, Dr. Lee, thank you for joining us today.
Thank you.
Okay, so first of all, would you be able to give us a brief overview of your research work?
Yeah, so this is research is about this vitamin K dependent gamma carboxylation,
Mm-hmm.
which plays a central role in the hemostasis. So this a post-translational modification that converts glutamate residues to the gamma carboxyl glutamate or GLA residues in the so-called GLA domain of several coagulating factors and anticoagulating proteins.
Mm-hmm.
And they're important because these GLA residues bind calcium to promote the proper folding of the GLA domain and its association with the phospholipid membranes. So this membrane association is required for the assembly of a functional enzyme complexes that either promote the coagulation cascade or prevent the excessive thrombosis.
Mm-hmm.
So we basically determine the cryo-EM structures, also did a lot of functional studies, try to figure out how this underlying process works, this particularly this gamma carboxylase,
Mm-hmm.
that how this enzyme works on the to catalyze this post-translational modification.
Mm-hmm. So thank you for that wonderful introduction, and it's very exciting and interesting work. So congratulations for that. So the role of the vitamin K dependent gamma carboxylation in hemostasis is important. I'm wondering which unmet clinical needs that motivated you to undergo this investigation.
Yeah, so modulating this carboxylation process through the vitamin K has long been used clinically
Mm-hmm.
and saved the countless lives from bleeding and clotting.
Mm-hmm.
In the newborns, so giving vitamin K right after birth is a standard practice to prevent hemorrhagic disease, which can be a fatal condition caused by insufficient carboxylation. And in the older adults, like reducing the carboxylation using vitamin K antagonists, such as warfarin, remains one of the most commonly used anticoagulative therapies.
Yes.
The warfarin targets like multiple coagulation factors, so therefore it has certain advantage over these new like direct inhibitors or NOACs. For example, warfarin is a preferred option in the patients with mechanical heart valves or severe kidney disease. So unlike the warfarin, the direct inhibitors have a drawback. They have a short duration of action,
Mm-hmm.
and therefore they exhibit more like fluctuations in a coagulation profile. But the warfarin also has its own problems. It has a narrow therapeutic window and a high individual variation. So, but the warfarin is targeting another enzyme in this vitamin K process. So we're our new goal is targeting this carboxylates now to develop better anticoagulants.
Okay. So, what exactly distinguishes the gamma carboxylation process from other translational modifications?
This is a unique post-translational modification that first it requires a coupling to the vitamin K, and also it occurs at the ER membrane, at the membrane interface. So the carboxylase itself is an integral membrane enzyme. So each like a carboxylation reaction, it's basically you are adding another carboxyl group on the glutamate residue. Okay, so each of this reaction requires a deprotonation of this gamma carbon of the glutamate by exceptionally like strong general base, which has estimated pK of 25, 28 that is rarely found in the biological systems. So the carboxyl has this remarkable capability of generating this super base from vitamin K in the lipid bilayer and also coupling this reaction with the glutamate deprotonation and the CO2 addition in the aqueous phase with the ER lumen.
Oh, okay. So that's very complex, but also interesting. So this enzyme, the gamma carboxylase, how does it recognize the diverse protein substrates when they're catalyzing multiple carboxylation events on a single polypeptide chain?
So this function of this GLA domain in the coagulation factors, they require multiple GLA residues to act together.
Mm-hmm.
They're typically ranging from 3 to 12 residues, depending on the vitamin K dependent proteins, which can be either coagulation factors or anticoagulation proteins.
Got it.
So depending on the sequence diversity despite all this sequence diversity on the different substrate proteins, the carboxyl recognizes through a propeptide, so which is generally located directly upstream of the GLA domain. So we basically determine the cryo-image structures of the carboxylates in complex with several substrate proteins.
Mm-hmm.
And this structure shows that different propeptides bind in similar conformation and form knob and hole interactions with carboxylase. So this high affinity binding brings adjacent GLA region for processive carboxylation. And the propellant is not released until all these target glutamate residue are converted to GLA residues. Okay, so that's how this processive carboxylation is working.
Okay. And what are the key findings from your study that advance our understanding of this unique catalytic process?
Yeah, so let's start with the propide binding. So this propide binding, let's say, is first that brings this GLA region for carboxylation.
Mm-hmm.
It also induce a global conformational change in the carboxylase
Mm-hmm.
that activates its catalysis. So this dual role of the propeptide ensures that carboxylation occurs specifically on the substrate proteins contains a propeptide. So another interesting observation we have that the from the carboxyl structure, we saw this enzyme have a large chamber that provides steric control over the substrate access and the positioning. So the substrate are too long may be excluded due to this overcrowding in the chamber. For example, in the coagulating factors,
Mm-hmm.
the gamma carboxylation occurs only up to the GLA domain.
Mm-hmm.
And if it's too long, it's not extended to a subsequent like EGF domain because this chamber has a certain like capacity in there. Uh, so back to the this other more complex chemistry.
Mm-hmm.
So we found that it's through this sequential deprotonation and the vitamin K oxidation, the carboxylase actually generate the free like hydroxy ion as a superbase that deprotonates the gamma carbon. So this superbase, since this superbase is free ion, right, it can diffuse through a sealed like hydrophobic tunnel. So that's kind of elegant way of solving this problem. How do you couple these two reactions together? Right? One is vitamin K oxidation, one is carboxylation across this membrane interface.
Dr. Lee, how do these data illuminate the molecular basis of bleeding disorders and broaden our understanding of non-hemostatic vitamin K dependent targets?
The mutations in the propeptide and GLA domain of coagulation factors are associated with bleeding disorders such as hemophilia B, thrombophilia, and factor 7 deficiency. The propeptide mutations also cause warfarin hypersensitivity in patients treated by warfarin. So our data reveals that the gamma-carboxylase binds propeptide at three primary sites. Of this, one site is critically linked to the hemophilia and warfarin hypersensitivity. So these specific sites present a very promising target for future drug development. The mutation in the gamma carboxyl alone causes other disease called vitamin K dependent carotene factor deficiency, which is a rare hereditary disorder characterized by spontaneous hemorrhage. We found these mutations clustered in several distinct structure regions that influence almost every stage of the catalysis, including the initial activation. Beyond hemostasis, the vitamin K dependent proteins participate in several non-hemostatic pathways, such as counteracting the inflammation and the apoptosis, modulating the immune response, and maintaining vascular integrity. They also regulate calcium hemostasis in the bone and soft tissues. In particular, we found that oxyocalcin, which is one of the most abundant protein in the human body, need to bind to another site on the carboxylase to enhance its proprietary binding. So this discovery opens the possibility of selectively targeting the carboxylation of oxyocalcin, which plays in the important role in the metabolism such as enhancing the insulin sensitivity.
So, on the other hand, do you think by targeting the gamma carboxylase would complement or improve our current approaches for managing thrombotic disorders?
Yes, the high resolution structures of gamma carboxylase we just saw provide the foundation for developing new anticoagulants.
Mm-hmm.
So our goal is to regulate this carboxylation pathway that can modulate multiple coagulating factors,
Mm-hmm.
similar to the mechanism of warfarin. However, unlike warfarin it indirectly affects this carboxylation by inhibiting another enzyme that supplies the vitamin K to the carboxylase. So now we can directly target this carboxylase, which may reduce the individual variability associated with the vitamin K levels. Also, the vitamin K, the carboxylase is an integral membrane enzyme. So membrane protein are known as major drug targets. So therefore, the carboxylate inhibitors
Mm-hmm.
could offer improved pharmacological profiles, such as the slower releasing of the drug and the prolonged stable action. So this may overcome the limitation of the current like direct oral inhibition or anticoagulants,
Mm-hmm.
which are short acting and they cause fluctuations in the coagulation activity.
Oh, okay. Thank you. So, Dr. Lee, I'm sure that your work will be contributing to the development of the novel target for the future therapeutic agents. So, congratulations on your work and thank you for being with us today. And I hope you enjoy the rest of your congress.
Thank you very much.